Hello everyone, welcome back again. So today I have come up with a topic that is DNA damage, or you can call it as mutation, that is very important for GATE Life Sciences as well as Biotechnology exam. So as you all know that GATE exams is approaching in the month of February. So for this we are coming to learn and revise all the concepts that is needed for GATE Life Sciences as well as GATE Biotechnology exams. So today I'm going to take DNA damage and mutations, where we are going to learn everything about DNA damage, its introduction, what are the factors involved, what are the types of mutations, what are the types of spontaneous and induced mutations, everything about DNA damage. So let's start with the video and understand the same. So coming to the introduction of DNA damage, that is mutation. So if you see about mutation, everyone knows mutation is a sudden changes that takes place in the genome of an organism. So these changes can be random. These changes can be random, it is not ordered. So it is a sudden heritable changes that occurs in the genome of an organism, and it results in the alternate form of any gene. If you know gene, suppose if there is any gene, let's say if it is AA, capital A, capital A. So if this capital A results in the formation of allele, so this is what called as mutation. This is what called as mutation. So let's say it is homozygous allele, that is homozygous. Capital A, capital A. And if mutation takes place, this results in the formation of allele. So this mutation is only responsible for the formation of allele, or you can say small a, small a. So it is a sudden heritable changes that occurs in the sequence of organism genome, which results in the formation of alternative form of a gene, and the process through which this mutation is caused, it is called as mutagenesis, and the result that is formed as a result of mutation. So here we are getting a small a, this is called as mutant. So mutant is an organism that exhibits a novel phenotype. But what are the causes of mutations? So the causes of mutations can be polyploidy, it can be aneuploidy or euploidy, it can be any chromosomal aberrations or any changes that is related to the gene level. Now what are the features of mutation? So make sure this mutation is a random process. It is not an ordered process. It can take any time or anywhere in the organism genome. So most of the time they are recessive, they are not dominant. Recessive means if both the genes are mutated for this homozygous AA condition, only then the mutation will be caused. But every time it's not the same. Sometimes the mutation can be dominant, but most of the time they are recessive. Mostly it is harmful to the organism, but when it is useful, it is serving as a source of variation. And you can see with this mutation only, this capital A is transformed to small a, that means allele formation. So this formation of allele only provides the ultimate source of evolution and is the source of variation. As you can see, variation in the organism genotype, or you can see in the mutant genotype. And also it results in the formation of allele. So this is about the introduction of mutation, what is mutation and what are the features of mutation. So coming to the reasons for mutation. So there can be of two types. The first is spontaneous mutation and the first is spontaneous mutation and the other is induced mutation. If you see spontaneous mutation as a mutation that doesn't need any mutagen from outside. What do you understand by mutagen? An agent that causes mutation. So this type of mutation, as the name only suggests, it is spontaneous mutation, means it doesn't need any mutagen from outside. But if you see induced mutation, means it is induced from outside. This type of mutation always needs mutagen from outside. It can be in the form of biological mutation, it can be in the form of chemical mutagen or physical mutagen. Means this needs mutagen for causing this induced mutation, and hence the name induced. So let's talk about spontaneous mutation first. So if we see, spontaneous mutation is further classified into three types. As you can see here, the first is errors in DNA replication, second is spontaneous lesion, and the third is transposition. So what do you understand by errors in DNA replication? So as you all know, DNA replication takes place both in prokaryotes and eukaryotes. But sometimes if errors is caught, so this is called as spontaneous mutation. So if replicated DNA is damaged by some means, so it is called as spontaneous lesions. So because spontaneously it is mutated, as no mutagens are included for this. So errors in DNA replication is further classified into point mutation or frame shift. The mutation can be at a single base, that is called as point mutation. This point mutation is also called as substitution mutation, because suppose if this is a DNA fragment, or let's say ATGC. Suppose from this a base, thymine base is replaced by another base. So this is called as substitution mutation, because one base is mutated and it is replaced by another. So this substitution mutation can be a transition mutation or a transversion. Transition means any purine is replaced by a purine, and we all know there are two types of purine bases in DNA molecule. One is adenine and the second is guanine. And pyrimidine, again they are of two types, that is cytosine and thymine. So transition means if any purine is replaced by a purine, or pyrimidine is replaced by a pyrimidine. Suppose this thymine, it is replaced by cytosine. So in place of T, C comes, and it will be present in DNA molecule. Then this type of mutation is called as transition, because here a pyrimidine base is replaced by pyrimidine. But what in case of transversion? A purine is replaced by a pyrimidine or vice versa, means pyrimidine is replaced by purine. So this is all about point mutation, that is a part of errors in DNA replication, or you can say spontaneous mutation. The second is frame shift mutation, means there will be an addition of sequences in the DNA that will actually shift the frame, that will cause the different protein formation. So this addition of sequences or deletion of sequences always leads to the shift in the frame. But this deletion of sequences, there are three examples that you have to learn, and that is important for exam point of view. So what all sequence will be deleted that will lead to diseases? So there is one disease that is what called as Huntington disease. So if you see Huntington disease, if repeats such as CAG, if CAG repeat continuously, it is deleted from the DNA. That will lead to a disease or a serious illness, that is what called as Huntington's disease. And if CGG repeat is deleted from the DNA, it will lead to another disease that is called as Fragile X syndrome. And if CTG repeat is deleted from DNA, it leads to myotonic dystrophy. So these examples are very important, and we have covered the frame shift and induced mutation you can say this addition and deletion under frame shift, and we have also seen point mutation, that is a part of errors in DNA replication. Now coming to the second type of spontaneous mutation, make sure we are understanding spontaneous mutation first, and this is the second subtype. What do you understand by spontaneous lesion? Spontaneous lesion means any naturally occurring damage in the DNA. Any naturally occurring damage that occurs in DNA is called as spontaneous lesion. Now again, this can be of three types. It can be depurination or depyrimidination, it can be deamination, or it can be oxidative damage. What actually happens in the depurination or depyrimidination phenomena? D stands for removal, means if any purine or pyrimidine base will be there in DNA, that will be removed. And if these bases will be removed, that site will be empty. Suppose this is ATGC, and if G is removed from here, this will create an empty space, and this is only called as apurinic or apyrimidinic site. It depends on the type of base that is removed. And here you can see this guanine is a purine base. So this will create AP site, which stands for apurinic site. This results in the formation of a purine base, or you can say apurinic sites, which results in AP site. Then the second is deamination. Deamination means removal of amino group from any base. And we know cytosine has an amino group in its structure, adenine also has, and guanine also has. What about thymine? Thymine doesn't have any amino group in its structure. So deamination of thymine is not possible. Why? Because it has no amino group. But here you can see 5-methylcytosine, which is an analog of cytosine. So this 5-methylcytosine can be be deaminated further to form any base. So let's see about the deamination profile of all these bases. So cytosine, when it is deaminated, it will form uracil. Very important. These all points are very specific and important. Cytosine deamination leads to uracil. If 5-methyl cytosine is deaminated, it leads to the formation of thymine. Adenine deamination leads to hypoxanthine, as well as guanine deamination leads to xanthine. So this is about the deamination event. What about oxidative damage? The name only suggests acid damage that occurs in DNA in the presence of oxygen radicals, that is called as oxidative damage. Now the examples of oxidative damage can be any reactive oxygen species, it can be hydroxyl radical, it can be peroxide radical, it even can be hydrogen peroxide. So all these molecules, they cause the damage in the DNA. So it mainly acts on guanine and it converts the guanine to form 8-oxo-7-hydroguanosine. So here you can see the oxygen is reacted with guanine so as to form an oxidative product, and you can see oxo in the prefix, and it forms 8-oxo-7-hydrodeoxyguanosine, which pairs with A. Now you know that G pairs with C. G pairs with the triple bond with C. But when this compound is formed, that is 8-oxo-7-hydrodeoxyguanosine, it pairs with A. This now pairs with A, means it will cause GC to TA transversion. This will cause GC to TA transversion because guanine that was about to pair with cytosine. So guanine was about to pair with cytosine, but now adenine will be bind to this 8-oxo-7-hydrodeoxyguanosine, causing the transversion. Why it is a transversion event? Because here you can see instead of cytosine, adenine is binding to this product. So the cytosine is a pyrimidine and adenine is a purine. So pyrimidine is replaced by a purine, means it is a type of transversion. So GC to AA transversion is happening here. Second, this oxidative damage also, or you can say this oxygen radicals react with thymine, that forms thymidine glycol, and if you see thymidine glycol, it is responsible for blocking the process of DNA replication. So this is all about the spontaneous lesion, where we have discussed 3 types: depurination, depyrimidination, we have also seen deamination event, and also oxidative damage. What is the third type that is coming under spontaneous mutation? It is transposition. And you know, for this transposition, the molecule that is involved is called as transposons, and this transposons is also called as a junk DNA that is responsible for the copy-paste mechanism. So this transposition is an event which means the shifting of DNA molecule, or you can say the sequences of DNA that is shifted from one position to the next. Trans here is stands for foreign, and position means position is shifted. Means if some sequences are present at some place for some time, that will be shifted to a new place. This is what called as transposition. Now this transposition can be a cut paste mechanism or a copy paste mechanism. So suppose this is A, T, G, C, G, C, T, A. Suppose if these sequences are shifted from here, so they will be cut from here and they will be pasted in a new position. Suppose they are pasted here. So this is called as cut paste position mechanism, and this is further going to change the open reading frame and will going to change the amino acid sequence and ultimately the protein expression, and the different protein will be formed that will be non-functional. And suppose if this copy is made, and the copy of this sequence is made, and now again it is pasted in this position only. This is called as copy paste mechanism, in which copy of the sequences is made and is pasted in a different position. Again you can see the open reading frame will be changed. Why open reading frame will be changed? Because we all know that one amino acid, it is encoded from a single codon, and every codon is a triplet. So this triplet will be changed and it will result in a different protein expression. So this is all about spontaneous mutation. Now let's come to the next type of mutation, or you can say reasons for mutation, that is induced mutation. As I've already mentioned, induced mutation stands for mutagen that is inducing to cause the mutation. So these mutagen can be of 3 types. It can be biological mutagen, it can be physical mutagen, it can be chemical. What do you understand by biological mutagen? Biological means something living that is changing the organism genome. That can be either viruses or transposons. So what are the examples of biological mutagen? It can be any viruses, it can be transposons, as we have seen for spontaneous lesions. The second is physical mutagens. Physical mutagens here relates to radiation. It can be any kind of radiation. It can be ionizing radiation, it can be non-ionizing radiation. So what are the examples of ionizing radiation? It can be alpha rays, beta rays, as well as gamma rays. So this ionizing radiation, as you can see, ionize. The ionizing radiation means it will ionize the DNA, and it will ionize, it will cause the strand breakage because all the bases will be ionized, so it will result in strand breakage. But what about non-ionizing radiation? Means they are not able to ionize the DNA, so in turn they will cause the formation of dimer. But what is the most frequent type of dimer that is formed in DNA in the presence of non-ionizing radiation? That is thymine dimer. So the examples of non-ionizing radiation is UV rays. Again, this is very important question for GATE point of view, if it is GATE Life Sciences or GATE Biotechnology, because the direct question can be asked in multiple select question like what is the example of ionizing radiation, and you will be given 4 radiation like alpha rays, beta rays, gamma rays and UV rays, and out of this you have to select 3 options like alpha, beta and gamma, because you can see the ionizing radiation there of alpha, beta and gamma, and non-ionizing is UV rays. So coming to the chemical mutagen. So this chemical mutagen is further categorized into base analog, deamination, that causes deamination means deaminating agent. The third is alkylating agent that alkylates the DNA, and the fourth is intercalating agent that intercalates or binds in between the DNA sequence and unwinds the DNA helix. So coming to the base analog examples. So this base analog example is 5-bromouracil and 2-aminopurine. So these 2 examples are very important under base analog. So if you see this 5-bromouracil, it is a base analog. It is a base analog of thymine. So this thymine binds to adenine. So we all know that thymine pairs with adenine. So this pairs with adenine. But ultimately, when this 5-bromouracil is formed, so ultimately it pairs with G. So how to learn all these types of transition? So here I will tell you one code how to remember all these types of transition that is happening here. So you have to just remember the code ATGC for all. ATGC in a sequence. So here you can see it is a base analog of thymine. So you just have to separate this ATGC by a gap. Separate AT and GC. So these both are separated by a gap. Now you can see here thymine, this is a thymine analog. 5-bromouracil means thymine is coming first. So thymine is coming first and A will come later. So again you have to reverse this position here also because you have reversed here. So CG. So it will cause TA to CG transition. It is very simple. And why transition? Because you can see adenine is replaced by guanine. So thymine, which was about to bind to adenine, now it is binding directly to guanine. So this is a type of transition. Again for 2-aminopurine you can do the same thing, but you have to learn this 2-aminopurine is a base analog of adenine. Sometimes this question is also asked, 2-aminopurine is a base analog of dash. So 4 options will be given. You have to select adenine from there. And you know adenine pairs with T. So this is simply AT to GC transition. It is in a sequence. A will pairs with T, but here 2-aminopurine is there that will binds to C. So this is a type of AT-GC transition. And again A was about to bind to T, but now it is binding to C. So T is replaced by C. So it is a type of transition mutation where pyrimidine is replaced by another pyrimidine. Deaminating agent. So here you have to learn 2 examples. The one is nitrous acid, the other is sodium bisulfite. And if you see nitrous acid, again it is specific for guanine, cytosine and adenine. Why not thymine? Again thymine doesn't have any amino group. So again guanine will be deaminated to form xanthine, cytosine will be deaminated to form uracil, and adenine will be deaminated to form you can say hypoxanthine. So again there will be a GC to AT transition because G that was about to bind to C means we are following this sequence. So GC in a same series to AT. So GC to AT transition, cytosine. So we are going in reverse direction. So CG to TA transition, it's very simple to learn now. And adenine, so we are again coming here. So AT to GC transition. Means adenine, if it forms hypoxanthine, that will pairs with C. So the last sequence means that base will pairs with this last base. And cytosine, it will lead to the formation of uracil. So that will pairs with A. And guanine, that will form xanthine, that will pairs with C. So that is different transitions we have learned. For sodium bisulfite, this agent, or you can say this deaminating mutagen, it is specific to only a single base, that too cytosine. So this is very important. You have to learn the sodium bisulfite is specific only for a single nitrogenous base, that too is cytosine. So again, cytosine will be converted to uracil, because here we are learning deaminating agent means the process of deamination. And ultimately this cytosine will be converted to uracil, and this will be forming CG to TA transition. Again, you can see cytosine will be forming uracil, that will pairs with A, and this is the CG to TA transition. So we have seen a deaminating agent and also base analog. Now we are coming to the alkylating agent, that is a third category under chemical mutagen. So here some chemicals are needed to mutate this DNA. Alkylating agent, the first example is EMS, that stands for ethyl methyl sulfonate, and the other is nitrogen mustard gas. So both these agent, or you can say alkylating agent, are responsible for adding methyl groups to nitrogenous bases. So they are specific for thymine as well as guanine. So this guanine is methylated at 6th position, and it will form O6 methyl guanine. It forms O6 methyl guanine. But when thymine is methylated, it forms O4 methyl thymine. It forms O4 methyl thymines. So here it forms O6 methyl guanine, and if thymine is alkylated, it adds methyl group at 4th position, that results into the formation of O4 methyl thymine structure. And again you can follow the same trend. A, T, G, C, you have to separate it by a gap, but G is coming first. So you have to follow this order. So it will be GC to AT transition. It will be GC to AT transition. Why transition? Because guanine was about to bind to cytosine, but now guanine is converted to this product, and this will pair with T. So this is GC to AT transition. What about thymine? So thymine, you have to again reverse this order. That will be TA, and again you have reversed this order, so you have to reverse this order as well. TA to CG transition. So here you can see TA to CG transition. So when students learn about all these things, they will easily learn about base analog, deaminating agent, all these things. But it is very difficult to learn about transition. What type of transition or transversion is happening here? And from here the question is definitely asked in exam. It can be any competitive exam, it is pakka asked. So that time it is very difficult to learn all these things. So that's why I'm making it clear, and I am just showing you a one trick that you have to follow, and that will be very easy for you to learn all these transition. Not even learn, you can just follow this trick and you will be able to make it out yourself. And thymine again there will be TA to CG transition, as we have discussed. So under alkylating agent you have one other example, that is hydroxylamine. So for deaminating agent we have seen nitrous acid, sodium bisulfite. For alkylating agent, one is EMS and nitrogen mustard gas, and the other is hydroxylamine. The formula for this is NH2OH. So you have to recall the formula. Sometimes they will not mention the word hydroxylamine, they will just write a formula and you have to deduce it. So it is specific for cytosine. So again it is specific only for cytosine nitrogenous base. So again the transition mutation will be ATGC. So you have to follow the reverse order, because the first is cytosine. It is specific for cytosine, which was about to bind to guanine, but now this cytosine is converted to some other product that will be binding to A. So this is CG to TA transition. Again, this is a type of transition mutation. So here you have seen in case of induced mutation, all the types of mutation that is happening is a type of transition mutation, not transversion mutation. Where we have seen the transversion mutation example, we have seen the transversion mutation example in the case of oxidative damage that was a part of a spontaneous mutation, and here guanine is forming one different product that shows a transversion phenomena. Now the last is intercalating agent. Agent that chelates or bind in between the nitrogenous bases. Here inter stands for between. Chelate means binding. ऐसा agent that binds in between the nitrogenous bases and slips off there. That's why it causes the unwinding of DNA helix and it separates the or increase the adjacent base pair ka difference. And the examples are proflavin, acridine orange, as well as ethidium bromide, that is very well known for the visualization of DNA fragments under trans UV illuminator. So we are done with the spontaneous mutation as well as induced mutation. Now we are coming to the types of mutation. Now if you see, there are 4 basic types of mutation. The first is silent mutation, the second is neutral, the third is missense, and the fourth is nonsense. What do you understand by silent mutation? As you can see in the diagram as well, this is TTC, that is the codon for DNA or the sequence in the DNA. This is mRNA sequence, that is AGG, that will encode for a lysine. This is a normal protein, or you can see a wild type where there is no mutation, no damage in DNA. But what if silent mutation takes place? So this silent mutation, it results in the change. You can see this is A. How A is caused? Because this DNA mutation has happened and this C is now replaced by T. So you can see now in place of T, A is there in the mRNA. Now again this A is responsible for forming the lysine. And you all know that lysine is an amino acid that has amino group. So it is a basic amino acid. So here you can see no nature of amino acid is changed. So silent mutation is a type of mutation in which amino acid is replaced by the same amino acid. Means if amino acid is replaced, it will always cause the addition of amino acid that was there in the wild type. So if amino acid is replaced also, again the amino acid will be same as the wild type itself, as you can see lysine here. Means there will be no nature change of amino acid and the nature of amino acid will remain the same as it is a basic amino acid. What about silent mutation? So silent we have seen. What about neutral mutation? Neutral mutation means change has occurred, but that was neutral in the DNA. One amino acid is replaced by another. So in neutral mutation, one amino acid will be replaced by another. So suppose here lysine is forming. So in place of lysine, arginine will be formed. But nature remains the same. Why nature? Because you know, again this arginine is a basic amino acid. So amino acid will be different, but nature will be same. How nature? Make sure we are not talking about the same amino acid. We are talking about the nature. Nature means it can be basic amino acid, it can be acidic amino acid. So for silent mutation, you have to learn the amino acid will be replaced by another amino acid sequence, but the nature will remain the same. So same amino acid will be incorporated, like lysine was there, again lysine will come and has the same nature. So amino acid and nature both are same. So here amino acid will be incorporated same and the nature of the amino acid will be same as it is a basic amino acid. But what in case of neutral mutation? Amino acid will be different. This time the amino acid you can see it is arginine, but the nature will be same as it is basic amino acid. What about missense mutation? Missense means by mistake something is incorporated. Means the amino acid will be replaced by another. Means amino acid will be different. Here I have mentioned same. Here again replaced by another. Here again another. So in missense, the amino acid will be replaced by another and this time the nature will also change. So suppose if there was lysine amino acid, so now here it will come threonine. Threonine means it is a OH group containing amino acid and OH group containing amino acid means the nature of the amino acid changes from basic to hydroxyl containing amino acid. So here the amino acid changes and along with this the nature of amino acid also changes. So both of these changes and you can see how the one parameter is different. Firstly both of them were same and now here amino acid is different. Now this is changed and here both are different. Amino acid and nature both is different. What do you understand by conservative missense mechanism? Means it is talking about neutral. It is talking about neutral mutation. It is talking about, as I have mentioned the examples for arginine, so it is talking about same. So if it is happening by a conservative mechanism, it is a neutral mutation, or non-conservative mechanism means it is talking about missense mutation only. What do you understand by nonsense? So nonsense means, nonsense, as you all know, sense means open reading frame, that will become nonsense, means it will not code for any protein, and the protein step, or you can say translation process, will be halted, or you can say blocked. Why? Because amino acid is replaced by another amino acid, and this time the codons will be a stop codon. As we all know, there are three types of stop codon: UAG, UAA, as well as UGA. And if any of these codon is incorporated in the open reading frame, that will stop the protein synthesis process, and it will shortens the protein length. Isliye this nonsense mutation is always related to the protein length shortening, because it incorporates the stop codon in between. As you can see here, this ATC that was there in the DNA sequence, this A is replaced by U. So UAG means this is a type of stop codon, and the protein synthesis will be blocked right there itself. So this is all about the mutations, where we have seen all things. We have seen the introduction, we have also seen the features of mutation, what are the spontaneous, induced mutation, we have seen different classification of spontaneous and induced mutation, along with the types of mutation, that are of 4 types. So now this is all about the topic. If you like the topic, do not forget to like, share and subscribe to the channel with Biotecnika. So very soon we are going to come up with another topic that is very useful for GATE Life Sciences or GATE Biotechnology. Just stay tuned to our channel. So this is all about the topic. Meet you in the next video. Until next time, this is your host Ria from Biotecnika. Keep learning. Take care.